First, I would say we're a computational photochemistry and photobiology group primarily is how we define ourselves. So what that means is we develop some computational tools and apply them to different problems in chemistry. Primarily what we're looking at is how chemicals and biological systems interact with light and what happens after they absorb that light downstream. How does it use that light energy in order to drive a chemical or a biological process? So this is done not in a wet lab. In other words, we don't actually do the experiments. We run the computer simulations. And these computer simulations rely on principles from quantum mechanics, thermodynamics, kinetics. So a lot of borrowed ideas from physics applied to solving these problems. So computer science plays a major role in what we do, because obviously we are a computational group. We have to rely on high performance computing in order to be able to do what we do. These are complex problems that require a high level of computational power. If we run them on a laptop, it will take forever. So we have to run them on giant nodes that are often going to be shared by many others, and we have to coordinate with each other in order to be able to run these calculations effectively. So I think that's, I would say, the biggest challenge is managing all of that and building the know-how. And certainly with students, when we do that, we learn as we go. We start out not knowing enough about each field, but then as time goes on, we pick up the knowledge in these different areas that is needed for our research and put it all together in order to make it all work. A lot of the research that we do relies on being able to have access to high performance computing and many computational tools that are available to us through, for example, Arctic. The main reason for that is we run different kinds of calculations, but for instance, we run quantum mechanical calculations that require diagonalization of large matrices. These kinds of calculations are going to require high computing power, powerful CPUs that are able to run these calculations, multiple cores on these CPUs to be able to run these calculations in parallel. And on the other hand, we also work with large systems like proteins. So these have many body interactions, all the atoms interacting with each other, and solving these kind of equations require not only maybe not solving complex equations in that particular case, but solving many simpler equations. And these kind of calculations require a different kind of infrastructure, for example, graphical processing units, in order to run more efficiently. So one of the main projects that we work on at the moment is understanding types of proteins, the mechanisms of proteins that respond to light. Those are called photoreceptors. There's surprisingly few of them, considering how important light is to organisms and their survival. We interact with light all the time. It regulates, first, vision is the most obvious case, but it also regulates our circadian rhythm and our day-night cycle. The proteins that are responsible for regulating biological processes in response to light, there's not that many of them. There are a few large families. Rhodopsins are the famous ones. These are the ones that we have in our eye that are responsible for vision. Phytochromes, for example, cryptochromes, bluffdomains. These are some of the larger families. We have recently been looking at what are known as light oxygen voltage sensing domains. These are proteins that are found in plants, in fungi, in certain types of bacteria, where they respond to light and allow some downstream biological process to happen. There's been a lot of interesting research in that direction, because you can take these proteins that are naturally supposed to do one thing and then install them as almost a kind of a light switch to activate a completely different function artificially. So for example, you can install that protein as a light switch that activates or regulates an enzyme or DNA binding. So there's been a lot of, I would say, exciting experimental work, no pun intended, in that area of light excitation and downstream regulation of biological processes from there.